Hi everyone, and welcome back to the second half of the genetic testing lesson. So let's continue with direct-to-consumer genetic testing, or those at-home genetic test kits. Direct-to-consumer genetic testing basically provides people access to their genetic information without having to involve a healthcare provider or a health insurance company. There are two types of genetic testings, or two categories. The first one is called ancestry testing or genealogy testing, and this can provide clues about where a person's ancestors might have come from and about family relationships. The other type of testing is testing for traits or health genetic testing, and these tests are going to test for the presence of specific gene variants for either harmless traits or even nowadays some genetic disorder. Some companies will test for only one thing, so some companies will test only for ancestry genes, and some will test for just health and traits, and others will test for both. So depending on what you want and how much money you're willing to pay, you can get a variety of different types of tests done. So to simplify, most of these tests use a structure called a microarray. And a microarray is a microscopic piece of often ceramic to which single-stranded pieces of DNA is attached. And the reason why single-stranded pieces of DNA are used is because these pieces of DNA are specifically chosen to be complementary to the code for the specific genes that the testing kit is looking for. So for example, if the testing kit is looking for the presence of the gene for wet earwax, then the genetic code for wet earwax would be found as one of these single-stranded pieces of DNA. And that way it will allow for the customer's DNA to find that mutation or that gene variant and attach to it if that person happens to have that particular trait. So a microarray actually has thousands of these single-stranded pieces of DNA called probes attached to it. The customer's or the patient's DNA, which is of course double-stranded, not single-stranded, because it is bound to a complementary section of DNA. So, for example, whenever there's an adenine, its complementary base thymine is attached, and the attachment is done by hydrogen bonds, which can actually be broken by heat, or the strands could be reannealed by the removal of heat. And the first step is to make copies of this DNA strand using a process called polymerase chain reaction, and then cutting the DNA by restriction enzymes. Then heat is added, which are basically going to then separate the strands, and then now the separated, cut up pieces of DNA are placed with the microarray. And this is where the process begins, because now wherever a section of DNA is found to be complementary to one of the single-stranded pieces of the customer's DNA, it will anneal or attach by hydrogen bonding. And as the heat is lower, this reannealing will happen. And what's interesting is that wherever the reannealing happens, a process will be added that will make this particular strand of DNA that has attached to the probe glow with a fluorescent marker. So ultimately, when the whole process is done, only the DNA probes to which complementary strands from the patient or the customers have been attached will be the ones that glow. Now, the process obviously can happen with thousands of these DNA probes at a microscopic level and would be impossible to analyze with the naked eye. So computers basically do the analysis. However, in order to visualize how this analysis is done, let's just simplify it by looking at what a very simple microarray might look like. And so a microarray might say, for example, have probes for the sequence with the mutation that you're looking for. Say, for example, you're looking for specific genetic mutations for diseases, and it would have probes that would be complementary to the sequence without the mutation, so the normal sequence that you would find in people without the disease gene. And so the areas that are purple would have been areas that are glowing in the dark, that are fluorescent, that show the presence of that particular gene sequence in the patient. So let's say, for example, this particular probe or couple of probes was added to check for the presence of Tay-Sachs disease. And so this particular microarray would have a probe for the mutated Tay-Sachs gene, which is an autosomal recessive gene, and it would have a probe for the normal, non-mutated, non-Tay-Sachs genes that could be found in people without the disease. So in this particular case, what do we have here? We find that there's fluorescent where the Tay-Sachs mutation is. So obviously, this patient has one of the recessive alleles for Tay-Sachs disease. But there's also fluorescent in the non-mutated probe, which means that this particular patient, because every person has two alleles or two copies of a gene, one from mom, one from dad, this particular person has a mutated copy and a non-mutated copy. So they are essentially carriers for Tay-Sachs disease. So this is how a carrier test can be done. And this next probe, because we would check for many disorders or traits at the same time, this next probe might be testing for sickle cell anemia, which is again is an autosomal recessive disease. Now in this particular case, the fluorescence is only found in the probe without the mutation, which means that this person did not have a sequence that was complementary to the mutation. So one of its genes would have been the dominant non-mutated allele and the other also the dominant non-mutated allele, both of them attached to the same probe. And so you would have an unaffected person. This person is not a carrier and does not have sickle cell anemia. This next one might have been testing for cystic fibrosis, another autosomal recessive disease, but in this particular case, only the fluorescent probe is found within the sequence with the mutation. So in this particular case, both copies of that patient's genes tend to have the cystic fibrosis alleles. So this person actually has cystic fibrosis. This would be a positive test for the disease. What about if we're looking for an autosomal dominant trait? Like say, for example, we're looking for the presence of the BRCA1 gene or the BRCA1 gene. In this particular case, the mutation is a dominant trait. And this particular person does not have the mutation. So they have both recessive alleles, which basically means they don't have the mutation. And so this person does not have a higher risk of breast cancer because they don't have the BRCA1 gene. And in this other case, let's say we're looking for Huntington's disease, which is an autosomal dominant disease. In this particular case, this person has only one copy of the mutation because it has its other gene sequence was fluorescent and it attached to the probe without the mutation. But unfortunately, because this is an autosomal dominant disease, it doesn't matter that there's only one copy of the mutated gene present. This is a person who will develop Huntington's disease, might have symptoms of it already depending on that person's age. So this is basically how probes are used in order to determine whether or not certain traits are present or not in a particular patient. One of the craziest applications of genetic testing is pre-implantation genetic diagnosis, or PGD. Essentially, through PGD, parents can test their embryos for certain genetic disorders or traits, and only those embryos that basically pass the test, those embryos that show to have no undesirable genetic traits, are allowed to be implanted into the mother's uterus and therefore are allowed to be born. And this type of technology combines both in vitro fertilization, or IVF, with genetic testing. So it starts with in vitro fertilization, which is a 7-step process, but the process is halted between steps 5 and 6 to allow for genetic testing. And so before the embryos are transferred into the mother's uterus so that they can grow into a baby and be born, the embryos are tested for genetic abnormalities. A cell from the embryo is removed, and that cell is then analyzed for chromosomal abnormalities as well as any kind of genetic mutations that the parents are looking for. And in Canada, there are really not many rules as to what traits can be tested for. The only rule that we have in Canada is that we cannot test for sex, or we can test for it, but we definitely cannot use sex as a determinant of whether or not an embryo is going to be implanted or not. However, in the US, there is no such rules. And in the US, parents can select for genetic abnormalities, but they can also select for gender. They can basically choose whether they want to have a girl or a boy and have only those embryos that are healthy of their selected gender be implanted. The other thing is that in both Canada and the US, there is actually no rule that says that the only things that can be tested for is genetic abnormalities. There is a lot of traits that we can now test for that have nothing to do with health and that have nothing to do with genetic mutations and genetic disorders. And essentially, there is actually no law that stops parents from choosing to test their embryos for any and all of those particular traits, other than perhaps the expense. Now, in the UK, for example, there are actually rules about pre-implantation genetic diagnosis that limits the number of hospitals that can provide the process and regulates which parents are approved for PGD based on whether or not they, number one, are only choosing to test for traits that are detrimental to the child's health, and number two, do not test for sex or gender. So that's the difference. Now, in Canada, we cannot test for whether the babies are going to be males or females, so let's take that off. So the process will then continue from there on. So the embryos are cultured after fertilization happens in vitro in a test tube or a petri dish usually, and the genes of the embryos are tested, and then only those that pass the test, this one and this one, get to be transferred into the uterus and hopefully can result in pregnancy, which can then result in healthy babies being born. Now, one ethical concern, of course, with this type of technology is that it could lead to designer babies. Of course, everybody wants to have healthy children, but nowadays when we can test for pretty much almost any trait, and as genetic testing advances, more and more traits will be able to be tested for. The rules so far do not stop parents or doctors to test for basically anything and then select embryos according to just some arbitrary decisions that parents are making as far as who they want their children to be and what traits they want their children to inherit. Okay, last we have the last type of testing, which is the one that tests for proteins, which is called newborn screening. And newborn screening basically is a type of test that happens in every Ontario and Canadian hospital and most hospitals throughout the world, that does not actually test for DNA at all, but instead is testing for proteins that are found in the blood of a newborn. And these proteins are often a result of some sort of faulty gene. So this may be a case in which proteins are looked for and to see if they're missing, or proteins that are misshapen are found, or proteins that are produced in too large quantities perhaps. And the amount of tests that are done in hospitals varies depending on the amount of money the hospitals and provinces in Canada or states in the US or countries are willing to put forth for this type of testing. This is a type of testing called screening. So that means that everybody is tested for. That means that this is part of public funding. So for example, in Ontario, we used to test for only 2 genetic diseases back in 2003, but by 2014, 29 diseases were being tested for, which is one of the highest numbers of tests that are being performed in Canada and one of the highest in the world. This is often done at birth. When a baby is born in a hospital, a nurse will come in within a day or two after the baby is born before they leave the hospital, and they will just take a little sample of blood from the baby's heel, from the foot, and then they will put drops of that blood in a special type of test paper that will collect the blood, and then that test paper with the baby's blood is sealed and is sent to a lab, and then a few days or weeks later the results are going to be passed on to the child's family doctor, and then if the tests come up with any sort of genetic condition, then to the parent. What's the point of this? Why test babies? Why not just wait until symptoms appear? And the reason for that is because oftentimes these tests are done for genetic conditions for which early intervention is key. if you catch it early enough. Oftentimes, if you catch it before even symptoms appear, the chances that the child is going to have a normal or close to normal life is going to be increased, or at least the chances of complications is going to be reduced. Let me give you an example here. There's a test for a disease called PKU, or phenylketonuria. And this particular test is one of the most important types of newborn screening that we do. So PKU is an inherited genetic condition that basically makes a baby unable to break down an amino acid called phenylalanine. Now, phenylalanine is an amino acid that is found in a lot of different foods that are high in protein. And when babies are unable to metabolize or essentially break apart this amino acid, the amino acid builds up to toxic levels. And the place where the amino acid builds up the most is in the baby's brain. And so PKU can essentially lead to developmental issues. A baby that was growing normally and was developing normally will suddenly develop brain damage and will lose all the progress that the baby has made. And the more protein the baby eats, the more phenylalanine accumulates in the baby's brain, the worse the condition gets. And essentially, the worse the brain damage gets. Fortunately, there's a very simple solution to PKU, and that's a diet that is free of proteins that contain high levels of phenylalanine. And so when a baby is diagnosed with PKU when they're born, before they are able to accumulate a lot of phenylalanine in their brain that will eventually cause brain damage, the test for newborn screening will have come back positive, and the parents will have been contacted so that they can place the baby on a special formula that is free of phenylalanine, because the breast milk will have it, and so will regular formula. But a special phenol-free formula can be given to the baby when it's an infant, and then as the baby starts to eat solid food, it will never eat any meat, any dairy products, any dry beans, nuts, or eggs, because those are high on phenylalanine. It can still eat cereals, fruits, and vegetables, but only in moderation and only certain types. So most of its diet is going to be supplemented by this phenylalanine-free formula, which will continue to be supplementing their diet even into childhood and adulthood. So it's not an easy disease to live with because you essentially cannot eat a lot of foods, but it is a simple way to prevent permanent and irreversible brain damage. And a person with PKU could be, say, for example, 20 years old. And if they have a steak, that steak will contaminate their brain. So the longer a person can go in their life without consuming phenylalanine, the better off they are. And the longer they're avoiding brain damage. So that's it, guys. That's it for our lesson on genetic testing. Bye-bye, guys.